All right, welcome back. Today we are going to be looking at how proteins and RNAs are transported into and out of the nucleus. So if you recall, the nucleus is a double membrane structure which has these complex pores that go through it, allowing a channel for passage of things in and out. Now, small molecules can move freely through passive diffusion, but larger macromolecules, such as proteins or RNAs, need to be specifically transported through the pore. And if you recall, the reason for this is in the center of the pore, we have this thing called FG nucleoporin, which is a long filamentous type protein, which is largely hydrophilic, except for these small hydrophobic patches. Those hydrophobic patches interact with other FG nucleoporin proteins, and it forms a gel-like material. So if the molecule that's needing to pass through is smaller, much smaller than the passages through this gel, then it can just easily transport right through. The more a molecule has to wiggle around to get through all these channels, the slower it's going to move through. And we have a way of facilitating getting larger molecules through this, and that is accomplished through interacting proteins which help transport it through. And we'll get to that there. But first, let's talk about how we decide what will go in or what will go out. We'll start off with going in here. So, proteins that need to be transported into the nucleus will have a amino acid sequence tag that is a part of its primary structure. So if you remember, primary structure of a protein is just the amino acid sequence. So here we see two examples of what we call a nuclear import signal. Okay, so here we have the T antigen, and you can see that this is a proline followed by several lysines, arginine. What we largely have here is a very positively charged cluster of amino acids, and this is what's acting as a signal to transport it through. Essentially, we have a transport protein which will recognize this signal and bind to it. And that other protein, which we call an importin, because it imports other proteins, will facilitate movement through this gel-like material. Here is another example of a different nuclear import signal. Here you can see that instead of it being one continuous tag, it is separated into two blocks. So here we have a couple of positively charged residues, followed by a stretch of 10 amino acids, which could be anything, followed by some more positive residues. So this is going to be the first example of um, using tags of a specific amino acid sequence in order to facilitate a job. And we'll see this occur again and again on different proteins. A lot of proteins will have a tag on the front indicating where it's supposed to end up. And this is just simply another example of that. This sequence is just telling it to be imported into the nucleus. Now, how this works is the protein that binds to the sequence has other binding sites, and those other binding sites recognize the actual FG nucleoporin gel material itself. So the important molecule will recognize this gel. So when it comes down here and interacts, it will actually hold on, giving it time to kind of work through. Now that interaction with this gel-like material is transient. It's a weak binding. So you can imagine that it's constantly binding and letting go, constantly binding, letting go. And by continually grabbing hold, it prevents it from floating back out backwards. And just over time, it'll be able to move progressively through here by letting go, binding to something next, binding something next, binding something next. And these cytoplasmic tails of the pore actually help facilitate this because they also bind the important protein. So if it were to fall out, it would simply bind to this and start its passageway back through. So now we have a tag on a protein that allows us to know that this protein needs to go into the nucleus. And we know that there's a protein that will allow it to transport through this gel-like material in the nuclear pore. So let's take a look at this mechanism. So here we have a cargo protein, and we see we have a nuclear localization signal. That doesn't need to be on any particular part of the protein, it just needs to be exposed. So it doesn't have to be at the front, it doesn't have to be in the back, it doesn't have to be in the middle. It could be any one of those, so long as it's on the surface and available to be seen by this protein called important. Now, there is a particular protein called important, but we're going to use important as a class of proteins because there are several members in this class, and they all recognize different nuclear localization signals. So what happens here is it recognizes the nuclear localization signal, they bind together, and if you recall, important has binding sites for these cytoplasmic tails, so it'll bind to that, stay close by here. Again, it's constantly binding, letting go. It also can recognize the Fg nucleoporin gel-like material, and it's recognizing it, binding it, letting it go, binding, letting it go, and sooner or later it can work itself down through. Once it's down on the side, we have this other protein called RAN, and RAN can bind GTP, or it can be bound to GDP. And depending on which one it is bound to, it has two different conformations. So when it's bound to GTP, it is in conformation 1, and it can bind this important molecule. When it binds that, it causes the cargo protein to fall off. So it breaks the association with the cargo protein. Now, this important GTP-RAN complex gets transported back out through, and in this case, we have both RAN and important, which can recognize the nucleoporin, allowing it to pass out through. Okay, so once it has passed out through, it will come into contact with this thing called RAN-GAP, or GTPase activating protein. So RAN can hydrolyze GTP into GDP and releasing a phosphate group, but it only does this enzymatic activity when it comes into contact with a GAP. So it transports through here, it comes into contact, it hydrolyzes, it changes into a second structure conformation. We can call that conformation 2, and that is incapable of binding important, so it'll kick it off. Now we have important free to continue the cycle to bring another protein in. Okay. Now, we need to replenish our stock of RAN GTP. Otherwise, we would simply run out of it and we could never bring important back out. So we have RAN GDP in the cytoplasm. It'll interact with NTF2, which is another member of this important class of proteins. and its job specifically is to import RAN GDP back through the nuclear pore to the other side. Once it's on this other side, we have a protein over here called RAN GEF, which stands for GTP exchange factor. And here what it does is when it comes into contact with RAN, it will cause the GDP to fall out and allow a fresh GTP to bind back in, which will of course change the conformation of RAN into its conformation 1, which will allow it to export important. Okay, now recognize here that now we've created another scenario where we have a lot of NTF2 on this side. NTF2 simply has its own export protein, which will transport it back out as well. And what this whole cycle really depends on is the fact that in one conformation, RAN can interact with important and move things out, but when it's in a second conformation, in the GDP conformation, it can't bind to that protein, and so it can only be re-imported back in. So the fact that whether it is bound to GTP or GDP is all regulated by two different proteins. Here we have the GTPase activation factor, and here we have the exchange factor. All we have to do is simply put a nuclear import signal on GEF, on the exchange factor. It'll get imported in and it will stay there. We don't put one on GAP, it will constantly stay outside, and these will act as the gatekeepers, allowing RAN to be cycled appropriately, and it will carry along its other proteins with it. So here we can see important brings things in, and then it gets transported back out through this RAN GTP, GDP type of cycle. Now, RAN GTP can not only transport the important molecule back out, but it can also transport other molecules as well. One of those molecules being the protein exportin. Exportin does what its name suggests. It can export things out of the nucleus. This is regulated by being bound to RAN GTP. So when exportin is bound to RAN GTP, it has a binding site for a sequence called a nuclear export sequence or signal. So a cargo protein that needs to be transported out will have this sequence, this export signal, which again is just a string of amino acids that are acting as a particular binding site for this exportin molecule. And in this case, the exportin molecule kind of acts as an adapter between RAN and the cargo protein. So the complex of all three, when bound to GTP, can be exported out as well. Again, when it moves out, we have this activating protein, which is sitting right at the exit of the nuclear pore. It will cause RAN to hydrolyze its GTP into GDP. That will cause the whole complex to fall apart. So not only will RAN fall off of exportin, but exportin will also release the cargo protein. Now we have the exportin protein, which can reassociate with RAN GDP in complex with NTF2. That will allow them to passage back through, and we can recycle the exportin, which can then move more proteins back out, and we can then take our RAN-GDP, make it associate with the exchange factor, which will make it drop the GDP and pick up a GTP and be able to repeat the cycle again. So you can see, just simply by keeping the GAP on one surface and the the exchange factor, GEF, on the other surface, we can regulate the process of several proteins being moved in and out. Okay. Now, again, there are several proteins which fall into this exportin category. It's not just a singular protein. There are several different kinds. And there are several different important proteins. Like, we could consider this NTF2 to be its own very specific important protein. Now, the other major thing that we need to export from the nucleus would be the messenger RNA. Messenger RNA has its own set of export complexes. So if you recall, we have messenger RNA gets modified in several different ways. It gets a 5' cap, okay, and it also gets a poly A tail. There are specific proteins which associate with that 5' cap and the poly A tail. So these proteins associating with the 5' cap and the poly A tail act as a maturation marker. So it just is indicated that this messenger RNA has finished its processing and it is now ready to be exported out. So the presence of these proteins bound to the 5' cap and the poly A tail allow this exporter complex to form. It will act in a similar way to exportin and RAN, in that it can bind the FG proteins that are in the pore, and it can allow the whole complex to work itself through the nuclear pore into the cytoplasm. Now, sitting right here at the exit to the nucleus is something called a helicase. If you recall, helicase is a protein that can move along a single-stranded nucleic acid. In this case, it's RNA. We've seen this before in DNA replication, helicase causing the two strands to separate apart. And it does it by moving along a single strand. So moving along a single strand here is essentially knocking off the exporter complex and releasing the mature messenger RNA into the cytoplasm. These proteins will then be recycled back into the nucleus using the mechanisms we've already discussed. Now, the last thing that I want to talk about with nuclear transport is the transport of this thing called snRNA. This is something that we hadn't talked too much about. This is one of the different versions of RNA that are out there. Remember, we have messenger RNA, we have transfer RNA, we have ribosomal RNA, but we also have something called small nuclear RNA, and these are RNAs that are involved in the splicing of messenger RNAs. So it can be involved in making sure the splices occur and also facilitate alternative splicing. But this is not just an RNA that does that. It also is associated with various proteins. So in this case, what we actually do is export the snRNA into the cytoplasm. In the cytoplasm, it'll associate with all of the proteins that it needs, and then we specifically re-import this whole complex back in. Otherwise, what we would have had to do is import each one of these proteins individually and allow this complex to form in the nucleus. Either method could have worked. The cell found this to be the most efficient. So here we have a specific protein which is transporting the small nuclear RNA out, and then we have another specific class of important molecule, which is specifically bringing in this snRNA protein complex, also which can be called snRNP, which stands for protein, back in, and then this will go to the splicing center in the nucleus, which is what the nucleolus is, that little dark region that you always see in a picture of a nucleus. All right, so here are the other members that are out there, or just key examples of various proteins that are involved in the import and export of various things. And you can see here we have important. It is responding to a particular amino acid sequence. This is the nucleoplasm signal. This is that bipartite signal. But there's another protein that would recognize the other signals that are out there. We have one that's specifically recognizing this snRNP complex. We have transportin, which is looking for messenger RNA binding proteins. Remember, there were proteins that bound that messenger RNA indicating maturation. Those are specifically transported in. Here is something that is specifically transporting in some histone proteins and also some ribosomal proteins. So you can see that there is a number of proteins in there, and similarly, there are a number of proteins that are involved in export. Okay, so now you should have a good idea of how to summarize how proteins and RNAs are transported into and out of the nucleus. Definitely focus in on those figures and be capable of being able to draw that out. Diagrams are one of the easiest ways of answering questions on my exams. So be able to draw out those figures and explain them. Now, I did want to take a minute here and talk about how this process of facilitating transport into and out of the nucleus can actually be used as its own regulation system for gene expression. So, as we've already learned in genetics, we have these things called transcription factors, which, when they come into contact with their binding site on the DNA, can lead to upregulation and more expression of the messenger RNA from that gene in particular. So, here we have an example of two different transcription factors. Here's NF-kappa B and FO4. They both have their own target genes that they will go to and turn on. The reason why they don't turn them on all the time is because they are regulated. And they're regulated here in two different mechanisms. So NF-kappa B has a nuclear localization signal, but it is covered by something called I-kappa B, which is another protein which associates with this. When the cell is ready to turn on the target genes of NF-kappa B, it's receiving a signal from the outside. So it is receiving a signal from outside of the cell. It will cause the phosphorylation and proteolysis of I-kappa B, which will make it go away, which will then expose the nuclear localization signal, allowing NF-kappa B to then be transported into the nucleus, where it can now do its job. Over here, we have a simpler solution. Here we have the nuclear localization signal being phosphorylated, which will essentially block any proteins from binding the site because it's now doesn't have the proper shape. or the proper charge. Remember that these localization signals are largely positively charged, and here we've put a big negative charge right smack on top of it. So here we have a protein which specifically dephosphorylates this in response to signals coming in from outside of the cell. Once that is dephosphorylated, it is allowed to be transported in. So here we can see we have a couple of different ways that we can regulate whether or not the transcription factors are even there to do their job, right? So recognize that the transcription factor itself is a way that we can regulate whether you make more or less of a particular protein. Here we can see we actually have a way that we can regulate turning those transcription factors on and off. And this is solely capable by the fact that we have all of the DNA inside of the nucleus. So here we have a physical separation, and the fact that we can regulate how things go through is giving us even more levels of regulation on when we turn on and off different proteins. And what you need to recognize is that differences between us and other organisms is not really due to differences in our genes. It's due to differences in when and where we turn on and turn off particular genes. So if you turn them on and off at a particular way, then you get a mouse. If you turn them on or off in a particular way, then you get a human. The genes themselves are largely identical. Remember that mice are 95% identical in their genes as we are to us. We're 99% the same with uh, bonobos. We are even more similar to each other despite how different we can look and how different we can act. So this is just yet another level that we can regulate when and where to turn on and off particular genes. So now hopefully you'll be able to explain how transport across the nuclear envelope can impact gene regulation or regulation of gene expression.